Hello, and welcome to this webinar titled Reducing Microbiological Risk and Using Quality Tools in the Pharmaceutical Industry, hosted by Biopharma Asia magazine and presented by Don Singer, Manager of Steriles Microbiology at GlaxoSmithKline R&D, and Jessica Razor, Product Manager at Charles River. My name is Stephen Edwards and I will be your moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the webinar. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first presenter. Don Singer is Manager of Steriles Microbiology for GlaxoSmithKline R&D, a GSK Fellow, and a Fellow of the American Society for Quality. Don has been a member of the USP Microbiology Committee of Experts since the year 2000. He is a former Malcolm Baldrige Award examiner and is a certified specialist microbiologist and certified pharmaceutical GMP professional. Don's career spans over 40 years of research, quality control, quality assurance experience in the pharmaceutical, cosmetic, and food industries. He is currently an adjunct instructor in the Biopharmaceutical Quality Program at the University of Maryland, Baltimore County, and an invited instructor to numerous global industry educational conferences and workshops. I'll now be handing over to our first presenter. Welcome, Don.
Thank you, Stephen. I'd first like to thank Stephen and the other editors and sponsors of Biopharma Asia for inviting me and giving me this opportunity. So let's begin our discussion. I'm going to be talking about a holistic strategy for microbiological control and how to pave a path towards better oversight using valuable tools from the practitioners who are responsible for quality as their primary job. Let's see how this develops, and as we discuss this strategy and bring more clarity to the thinking and learning aspects of developing and manufacturing drugs so that they're safe for our patients. We're going to be discussing quality, we're going to be discussing decision-making, contamination control, also analytical thinking, and other principles that are important for microbiologists in pharmaceutical laboratory and manufacturing roles. Now, microbiologists in our industry are quality specialists. Simply put, our mission is to ensure our products are safe to use. Now, we understand the variability of living organisms and trying to share this understanding with others. Yet the promise of microbiological safety is never guaranteed and is rarely proven. So the best that we can do to overcome this variability and the unknowns is to improve our understanding of the materials, the process, and the environment in order to make appropriate decisions to build contamination control assurance. Part of the strategy to build assurance is to understand risk. We've been provided industry-specific guidance to learn and use principles of risk. These are written by an industry collaborative, the International Conference of Harmonization, or ICH, and they define as well as guide users through a process that recommends a quality management system and what the risk management principles are. What is missing is the detail about how, and what are the best tools to accomplish this risk-based approach. So from the field of quality science, we have a boatload of tools that can be used. The intent is this. Quality tools have a number of uses. They can be used to measure, they can be used to gather information, and gather that information in an organized manner for further analytical use. They can be used to gather and organize data for analysis, or just for analysis in many different ways. The key point for using quality tools is to help make reliable, appropriate decisions and guide process improvement. A higher-level tactical approach is the use of DMAIC, or the DMAIC approach from a Six Sigma strategy. As you can see on this slide, the acronym has a simple foundation for a stepwise approach to quality improvement. Let's walk through these steps. Design can be related to product, process, materials, and facility. Measure is the generation of data to support the design, a way to monitor, and needing to monitor at a frequency that's appropriate for the measurement. Analyze is, of course, a key step. You need to make meaning from the data. Improve is the direction for changes that will lead to robust quality. And control is the ongoing step to ensure consistency for quality, which will lead to either another round of design change or maintenance of the current process. The MAIC is a cyclic cycle in steps and ongoing to ensure continuity. Six Sigma is a strategy for continuous improvement that will fit almost any process improvement situation. So, this is a quality strategy, Six Sigma, with a tactical approach. Yet the most critically important quality user is the microbiologist. Using information, knowledge, and data to better understand how to control a process and assure product quality can be accomplished by an experienced, well-learned quality control microbiologist. Microbiologists must play an active role. Use of all the human senses is part of an active microbiologist's approach to quality control. I cannot stress how significant this is. Seeing how a process is designed or how a process performs testing is accompanied by the knowledge of the process and procedures. An active microbiologist will be the foundation of a successful microbiological control strategy. And I'll return to this principle a little later. Microbiologists must influence control at each step of the product life cycle to develop, to manufacture, and to distribute drugs to the patients with the utmost safety and expected efficacy. We must protect the patients. How do we do this? We follow a holistic thinking process to improve our understanding of influences and impact that microbiology has on manufacturing and stability of drug products. We design drugs and processes to ensure a product meets the characteristics for efficacy and safety and will meet the expectations of customers. This is quality by design. Quality must be designed into products intentionally. Quality cannot be tested in. Tested is only a measurement, it's not a control. This is often misunderstood. Analytical testing for product quality attributes, especially when performed as a release test, is only a measurement of the effectiveness of controls that we design. Now, quality by design is a better approach to dealing with variability and unknowns of microbiological quality. As I mentioned earlier, we deal with variability all the time. Possible contamination is often heterogeneously distributed. Most of our tests only indicate use of very small samples. When we use media, it's not able to recover every organism that's present. If we use non-growth-based methods, we still have to determine specificity limitations. So we have inherent limitations to our test measurements. Thus, having a robust design for product and process is much more dependable. Let's talk about the key steps at a high level that require microbiological controls. We have the design of the formulation, which includes the active ingredients and excipients, and the design of the process. The manufacturing facility should be built for fit for purpose of the type of drugs being manufactured. The infrastructure of the facility is a significant contributor to controls. Primary packaging as part of design, along with secondary packaging, where appropriate, can add protection to the product. A product must remain stable with package integrity during its shelf life and distribution. How a product is used with the patient or by the patient must be understood to ensure nothing will contribute to a loss of microbiological safety. In order to build a better understanding of each of these product life cycle steps and process controls, we use risk assessment. Using a Six Sigma strategy, as I mentioned earlier, as the MAIC, evaluation can be performed for each step. As we can use quality tools and become confident with their use, the Six Sigma strategy will work well for us. Let's look at some of these quality tools and examples of their use. Product design is one of the first critical steps in drug development. Microbiological control aspects of this design phase are dependent on those parameters that inherently carry or subject the formulation to microbial contamination. One way to look at design is through evaluating the drug formulation with an Ishikawa diagram, also known as the Fishbone diagram. I'm sure you've seen this before. It's been commonly used for root cause analysis, but it works just as well in early evaluation and assessment of microbiological control parameters. As you can see in this diagram, inputs are indicated. There's excipients, dosage form, the active ingredient or API, and diluent, along with controllable parameters of each input. And while you assess inputs for formulation, you can also use a decision tree. This one happens to originate from ICH Q6A, which is the decision tree that can help assess the need for microbiological testing of material inputs of a formulation. Somewhat similar to the use of HACCP flowcharts, decision trees can help simplify an evaluation process. Another type of quality chart is one that identifies level of risk based on characteristics of a formulation. Here you can see the different types of final dosage forms, in other words, how a drug is administered, and where they fall regarding to risks to the human body. Parenterals are injected directly into the bloodstream or blood-enriched muscle, so no body protection is in place. Thus, it carries the highest risk of dosage form. On the other end, tablets and capsules are taken into the gut, which has numerous body protective mechanisms, thus much lower risk. Looking at a broader view of product manufacture, the fishbone can also be used to indicate what to evaluate in the controls for manufacturing inputs: formulation, process, environment, and personnel. Let's talk first about process. Process control is an engineering and validation arena, but microbiological issues must be realized and evaluated up front. The only CGMP required written record of a process is the batch record, but it may not be as helpful as you need. I like the Gemba approach, or walking the process in the actual facility. This is what an active microbiologist role actually is. If you can do this, by all means, it's the best option to start. Then follow it with developing or getting a flow chart from the process engineers to indicate each step and the critical parameters of each step. If you can create your own flow chart, by all means do that. It will be simpler to follow and use as a good reference tool. You can always improve it or add to it with discussions with the manufacturing operators or engineers as you learn more about the process. I mentioned HACCP before. HACCP, or HACCP, stands for Hazard Analysis of Critical Control Points. It is a thorough, easy-to-use risk assessment program that I have found to be much more relevant than an FMEA process. Learning how to use this tool includes defining the control points and the critical control points, along with learning what type of measurements are being taken in order to determine the ongoing effectiveness of each control. Each step in a manufacturing process, end-to-end, should be assessed for microbiological control. It doesn't matter what the dosage form will be. Each input can have an impact on product quality. Formulation steps, intermediate process steps, and the finishing operation, each can have impact on microbiological quality as it relates to the patient, the customer, and the product. In this flow chart, I've combined the HACCP critical control points with the process flow and the types of measurements that occur at each critical control point. This way I can evaluate data, go back to the process flow to determine where to take more samples or look for a change in the process that could have caused the trend that I've seen in data analysis. So the testing points are on the left in green, and the critical control points are identified on the right. When I mentioned FMEA earlier, I was referring to another tool used for risk evaluation, failure modes and effect analysis. This tool can take the process flowchart, fit it into a spreadsheet format, and then you perform a stepwise evaluation of the possible failures that can occur relevant to microbiological control. Each step is evaluated for severity or impact on patient or product. It's evaluated for the ability to detect a failure and the frequency that a failure has occurred or may occur. These risk parameters are rated and a risk level is calculated. The use of FMEA is often to determine where priority should be given to develop mitigation to prevent a failure of a process that can lead to a loss of microbiological control. Now, anytime data is generated, organizing that data in a format to evaluate clearly is important. Analysis of microbiological data is always difficult and confounding, yet the quality tools used for organizing data of any type can be very useful for evaluating the data. Here's an example of a bar chart, or an X-bar ARP chart, indicating the number of plates with growth from a different sample location at a point in time prior to any hold time of a material. Now, compared to that, this is a follow-up bar chart that follows the evaluation of the same samples taken after a hold time or storage period. This allows for a more clear picture of what happened from 0 to 8 hours of hold time. As you can see, the graphics changed. We're all scientists in microbiology, and sometimes we have the desire to develop a way to compare results that are difficult to evaluate. So experimentation is very common. Designing experiments allows us to test different conditions, separately or in combination, and analyze the data for clarity. Design of experiments is considered a quality approach across all arenas of quality. Now let's go back to our fishbone diagram. We see that there are two other key areas of manufacturing risk evaluation, the environment and the operations personnel. Let's look first at the environment. The common measurement approach for our environment, besides looking at temperature, humidity, and design, is monitoring the air passively, actively, as well as evaluating surfaces and personnel gowning. Now, with that data that we generate, it should be looked at for trends. A statistical process control is one means of analyzing data for trends. The tools we use for this type of analysis include run charts or X-bar R charts. Whatever type of data you want to analyze in this manner, there should be a benchmark that you want or have as an indicator of a process in control, and that way you can use that for ongoing comparison purposes. Now let's talk a little bit about personnel. Quality tools for personnel can help make decisions as well as help enhance training and assessment of skills. One of the most important parameters relating to microbiological quality are competency and skills and knowledge of the personnel. Now, we always need to have oversight of competencies and the training and testing of personnel for those competencies. This is something that relates directly to CGMPs, a requirement to be adequately and appropriately trained to operate in a pharmaceutical environment. We can use quality tools to enhance our ability to perform microbiological analysis and decision making. Here is one example, and it's a fault tree diagram or chart for following a positive sterility test result to determining a potential root cause. And this is a competency chart for individual laboratory personnel. It helps indicate where they have inadequate training and skills to perform certain activities for a microbiology laboratory. This could help identify areas for improvement or advanced learning. As you can see, it's broken down into different skill levels. Each employee is identified by letter, and then N means they're not trained, and star or asterisk means that they are trained. And you can generate and develop these tables in any way that you can that becomes useful for your own individual location and use. Here is another competency chart, and this one's for tracking skills and training for sterile operator manufacturing activities. It's set up the same way, except the skills obviously are different. And this can be used in microbiology, and it can be used in other types of manufacturing roles as well. So, to kind of summarize our discussion, all of this broad-based strategy for microbiological control is helpful. We have quality controls, we have quality tools that can choose to help us in developing a better understanding and help designing a better process to ensure high quality of our products. An active microbiologist will ensure success in this strategy. This is how we build quality into our products. Now, also on the next slide, and it's also found in the additional reference section for this webinar, are some references that provide more insights and examples, as well as options for other quality tools. So feel free to look at these when you have an opportunity and come up with other tools that may be useful in your daily jobs. And feel free to manipulate and use these tools in whatever manner that they can be helpful in helping you put in place well-designed and robust microbiological quality control. Thank you.
Thank you, Don. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Jessica Razor has been with Charles River for over 8 years, most currently in the role as Product Manager of Accugenix Services. She has extensive laboratory and R&D experience and has worked with manufacturing and supply chain management. Jessica has a BS in microbiology and MS in cell biology and molecular genetics, and a passion for science and patient safety. I'll now be handing over to our second presenter. Welcome, Jessica.
Thank you so much. Today I'll be talking about reducing risk with your microbial identification. So Don just gave a great overview of quality by design, the idea that we design quality into the entire manufacturing process. Those same principles can be applied to your environmental monitoring program. An effective EM program has several benefits. It ensures that a state of control is maintained, identifies areas for continuous improvement, reduces process variation and lead time, and assists in contamination investigations. So with such an important role, how do we design quality into our environmental monitoring program? One component that is often overlooked is your microbial identification system. As a microbiologist, it is critical that we understand the limitations of your system and evaluate the risk and quality associated with your ID platform. Now, the first part of my talk will describe different ID methods and their pros and cons, and then concludes with the operational impact of those IDs. So there are a number of different methods that can be used for microbial identification, and each method has a number of commercial systems. On the left, you can see the flow of information in the cell. Information is inherently stored within the DNA, which is then transcribed into messenger RNA, which is then translated into protein by the ribosome. Those proteins are then expressed in a variety of ways. And at each one of these points, there's an available identification method. Genotypic IDs are done through DNA sequencing. Proteotypic IDs look at protein distributions within the cell, and phenotypic IDs look at protein expression through metabolism and biochemical characteristics. Now, under each one of these methods, you can see some of the commercial systems listed. As you move away now from where information is inherently stored within the cell's DNA, out through protein expression, there is a reduction in accuracy and reproducibility for those ID methods. We'll take a closer look at each one of these methods to understand the advantages and disadvantages, system limitations, and the risks then that they apply to your system. So as I mentioned earlier, phenotypic systems differentiate organisms based on phenotypic attributes, such as morphology, biochemical, and physiological properties. Because they are growth-based, they require a live, healthy organism, and it often takes 1 to 2 days to get a result, sometimes even longer. Systems like API strips, Vitek, and BioLog use a series of biochemical tests which are read based on color change, turbidity, and precipitate formation. Some of these test results may have subjective interpretation of test results. Now, is that red or is it orange? Is that cloudy? And thus, there can be a higher rate of inaccurate or inconsistent identification. You may have heard of the 4I method. This is where 2 technicians have to sign off on the data interpretation due to data integrity concerns. And this doesn't always work. So let me tell you a quick story. My daughter wanted a pet when she was younger, and we compromised by getting a fish. Now, it's probably not what she was looking for, but that's what we said we're willing to do. My husband and I are both scientists, and we were trying to read the pH and nitrite strips when we were setting up the fish tank. It was not as easy as you would think to read those strips, and there was actually a lot of debate about whether or not that color was yellow or orange. In the end, we must have actually read the strips wrong because we had to have a fish funeral a few days later. Now, while these results were tragic to a 5-year-old, imagine what the consequences of a wrong ID in a manufacturing environment. There are also a limited number of tests as part of these commercial systems, so it can be very hard to differentiate closely related species. If you even end up with an ID, often it'll be a genus call instead of a species-level ID. Now, many people do like these systems because of their perceived ease of use. Many are automated and require little operator training. However, these systems often require ancillary testing like gram stains, whose accuracy can really depend upon the technician. We did a study a few years ago to assess the error rates of gram stains, and we found that error rate can vary, depending on the species, anywhere from 0% to 80%. Overall, it averaged about 20%. If your gram stain is wrong, often the ID will be wrong, or you'll end up with no ID and the test will need to be repeated. And then finally, these systems often have a limited library and are focused on clinical bugs, not necessarily organisms found in a manufacturing environment. If an organism is not present in the library, you cannot ID it accurately. Proteotypic systems are becoming more and more popular in pharmaceutical and consumer care environments. These systems are better known by their commercial names, such as the Bruker Biotyper, Vitek MS, and Accugenix AccuPro ID. The IDs are generated by a multi-toss mass spec, which stands for Matrix-Assisted Laser Desorption Ionization Time of Flight. A little bit of a mouthful. The organism is added directly to a small stainless steel target plate overlaid with an organic matrix that lyses open the cells and then forms complexes with the exposed proteins. A laser fires down and ionizes those complexes, which break off and then travel up the vacuum tube, where they are then detected based on their mass and charge. The resulting protein spectra is compared to a library of known spectra in order to make the ID. Now, proteotypic systems have very strict requirements for age and temperature of the organisms tested. Cultures need to be at least or less than 72 hours old and not exposed to the cold. This is because many of the proteins detected are ribosomal proteins, which begin to degrade with age and cold. So this means that you must have a live and healthy organism that is capable of growth in order to identify it with this system. Although the sample prep looks very simplistic, there's actually technique involved as to how the sample is spotted with both a direct smear and ethanol extract. There are also limitations as to how well the system can separate out closely related species, so additional tests may be required to get a species-level answer. Also, these systems were originally designed for clinical settings, so the libraries may be limited and not relevant for manufacturing. This is particularly relevant for fungal samples, which are of increasing regulatory concern. And finally, you have to consider how you'll interpret the results. Most commercial systems give you a match score, so you'll have to determine how to interpret that score and what level of accuracy you're willing to accept. The final ID method is genotypic. These are identifications based on the DNA sequence of the unknown organism. Sanger sequencing can be very labor-intensive and requires some technical expertise. So first, you have to start by isolating the genomic DNA from a single pure colony. Then, PCR is used to amplify the gene of interest, typically the 16S rRNA gene for bacteria, and then either the ITS or D2 region for fungi. The PCR product then undergoes cycle sequencing to incorporate fluorescent tags at the ends of the DNA fragment. These fragments undergo capillary electrophoresis, where the fluorescent tags are read base by base and results in an electropherogram. A data analyst examines the raw electropherogram, generates the consensus sequence, and performs a phylogenetic analysis in order to identify the unknown organism. I'm already tired just reading that out, all the steps involved. So DNA sequencing is considered really the gold standard for identification. But there are many things to consider before implementing one of these systems. First, as you probably noticed from my rather simplistic overview, sequencing can be very labor-intensive and very technical. A robust system has to overcome variations in DNA concentrations, lot-to-lot variations of reagents, and differences in instrumentation. Once the DNA sequence is generated, then you have to determine how you will handle the DNA analysis and interpretation, which will directly impact the accuracy and reproducibility of the ID. There are also concerns about which library or database you should use to match your unknown sequence, and this will also impact accuracy and to ensure your organism is taxonomically correct. This slide summarizes the characteristics of each ID method I just reviewed. DNA sequencing is the most accurate method, whereas phenotypic IDs are the least accurate. Sequencing can ID the broadest range of organisms, but it also requires the most operational skill. MALDI has the fastest time to result, but a high capital cost. So as you can see, each company really needs to weigh each one of these factors when choosing their ID method, and they also need to understand the limitations of each method as it will impact their environmental monitoring program. Keep in mind that within an ID method, not all commercial systems are created equal. This table is a little bit of a generalization, and there may be variances within each category. And also, this table does not include additional considerations of insourcing versus outsourcing IDs. Now, why does this even matter? Once the limitations of each microbial ID system and method are understood, the impact and risk to your EM program can then be truly assessed. This table shows some examples of ID results when the same culture is run on the MALDI and then also with API strips. The IDs were confirmed with sequencing, so we are able to know which answer is actually correct. In each case, the phenotypic API system incorrectly identified the organism, whereas the MALDI correctly ID'd the organism. This table describes each organism with a risk group as defined by the World Health Organization. You can see that risk group 1 means low individual and community risk. It's an organism that is unlikely to cause human disease or animal disease. Risk group 2, however, which falls under the Bacillus cereus, this is a pathogen that can cause human or animal disease, but is unlikely to be a serious hazard to laboratory workers. Laboratory exposures may cause serious infection, but effective treatment and preventative measures are available and the risk of spread is limited. What would this mean, though, if you recovered Bacillus cereus within your manufacturing environment? Would you be satisfied with an API answer of Bacillus species, or would you truly want to know a species-level answer? Now, again, how would an inaccurate ID truly impact your manufacturing decisions? Let's take also a look at the last one. The API strips incorrectly ID'd Burkholderia cepacia. This is particularly relevant in this day and age since the USP is writing a dedicated chapter on BCC recovery given the high number of product recalls. So depending on what product you were manufacturing, an ID of Burkholderia cepacia could have you scrapping an entire lot of products and undergoing a very lengthy and expensive investigation and remediation. And all of that just because of one inaccurate ID. Now, here's another example of different identifications, but these are a result of commercial system differences. These organisms were both identified using proteotypic methods, but we can see different results based on the libraries of the commercial system. So the AccuPro ID is Charles River's proprietary library, and then on the right is the Vitek MS produced by Maru. So remember that proteotypic systems generate protein spectra that is compared to a library, and an algorithm is used to determine a match factor. If an organism is not in the library, it cannot accurately be ID'd. So this slide really highlights the differences between Charles River and bioMaru's library. The Vitek MS has a smaller library that prevents identifications of these listed organisms. And again, why does that matter? You have to have an accurate ID in order to fully assess the impact a bug can have on both your product and your manufacturing process. Characteristics of your organism can help direct your investigation. Remember, you are making decisions based on your ID, so accuracy is truly paramount. Now finally, you always have to consider the risk to the patient. Is an inaccurate or irreproducible ID truly worth the risk? Are you confident enough in your manufacturing process, your EM program, and your ID system that you would administer your own product to your own family? Now, this is a special slide for me. That's actually my daughter in the picture. She has a number of orthopedic issues that requires many procedures and surgeries, and those are her x-rays. They look at the before and after of when her magnetic rods were implanted due to scoliosis. I share this because I really want everyone to remember that patient safety is not a catchphrase. Patients are real people who use our products. They are parents and children and spouses, and our goal and our priority should always be patient safety. Finally, tracking and trending is a very valuable tool that allows us to move from reactive quality control to proactive quality assurance. Tracking and trending allows us to identify problems before they're too big, allows us to see changes in the manufacturing flora, monitor IDs from action and alert limit, identify targets for process improvements, and measures the effectiveness of implemented process improvements. The value and importance of tracking and trending is increasingly recognized by regulatory bodies, which is especially evident in the EU's proposed Annex 1 revision. However, again, the benefits of tracking and trending can only be realized if the information is actually accurate. And again, that's going to rely on accurate and reproducible IDs. So to sum everything up, an effective EM program allows the manufacturer to demonstrate a state of control, identify areas for improvement, respond proactively to changes in the environment, and is key to an overall contamination control strategy. A key component to this effective EM program is accurate and reproducible microbial identification. It is imperative that the limitations for each ID method be understood so a true risk-based approach can be applied to manufacturing decisions. Accurate and reproducible identification depend on the ID method, data analysis, data interpretation, and the library being used. There are also differences between commercial systems that use the same ID method. Tracking and trending bioburden in IDs are necessary and helpful for making proactive decisions. So quality by design is well accepted for manufacturing, but needs to also be implemented in the microlab. Your ID system is a critical tool that deserves serious consideration because decisions are being made on those results, and the risk to the patient has to always be considered because patient safety cannot be compromised. This here's my contact information if anybody would like some additional information or has any questions. But at this point, that concludes my section, and I'll turn it back to Stephen.
Thank you, Jessica. Now we will begin the question and answer segment of the webinar. And once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now, our first question. Why do you feel that HACCP is a better tool to use compared to FMEA for risk assessment? Don, I believe this is for you.
Okay. And that's a good question because a lot of people are very familiar with FMEA, and only a handful are familiar with HACCP unless you've been in the food and cosmetic industry. My feeling is that HACCP is a active, ongoing evaluation tool. You identify critical control points. Each critical control point has some type of measurement being done, and you're tracking and trending the measurements from those points. Whereas FMEA is a tool that has been historically used in other industries as a preventative type evaluation. In other words, here's the process, what kind of failures can happen, and what are we going to do in order to prevent them from happening, more so than the other side of what's actually going on now, where a HACCP tool works a lot better, at least from my perspective.
Thank you. Our next question is, do you think that a microbiologist should have input into raw material supply assessments? Don, I think it's you again.
Okay. I mean, simple answer to that is definitely yes. The microbiologist, in a lot of cases, in a lot of roles, has a tendency of doing the job in the lab or in the manufacturing facility as they're indicated by the job description. But what happens is all of these other inputs that come in that can affect microbiological quality have a lot of preventative measures that need to be taken into account on the outside, externally, that we have to deal with internally. So the only way that we can do that is by using a microbiologist as a subject matter expert to work with the suppliers and deal with what controls they may or may not have in place and help to enhance that process better by understanding that better and what controls are or aren't in place, so that when we get the raw materials, we understand if we have to do anything more with them in order to provide safe microbiological quality.
Thank you. Moving on, what ID system would you recommend for routine environmental monitoring samples? Jessica, would you like to answer this?
Sure. So as you can see from my presentation, I put such an emphasis on accuracy. And so with that being said, the most accurate ID method is going to be DNA sequencing. So I recommend first DNA sequencing. Many companies, however, are hesitant to spend that much money on DNA sequencing because it is more expensive than some of the other ID methods. If cost is truly a concern or a limitation for your company, then I would suggest a proteotypic answer by using the MALDI, as this will be your second best, more cost effective, but yet still have a high level of accuracy.
On the back of that question, has Charles River implemented any of these quality by design tools in your company?
That's actually a really great question. So we really strive for continuous improvement, which is, a main pillar of quality by design. We also really strive to implement theory of constraints and lean manufacturing in order to minimize variation and increase accuracy. We try to prevent errors before they even happen. So one of the best ways for us to do that is through implementation of our LIMS system. Accugenix has a custom LIMS that allows us to control all of our personnel training, all of our equipment, all of our reagents and consumables, as well as our samples and process flow. So let me give you an example of how this works. We do all of our batch record documentation electronically through the LIMS. So if an operator was going to be performing PCR, before they even started, they would, everything's labeled with barcode, so they would scan their samples that they were ready to do that step on, PCR. They would scan their equipment, in this case a pipette. They would scan their PCR mix, as well as their own operator badge. Our LIMS system will go through and validate all of that information. It's going to ensure that it's the correct PCR mix for those samples, that it's been released by quality, and that it's within the expiration date. Our LIMS system is going to validate that that pipette is supposed to be used for that step in the process, and that the pipette is within calibration. It's going to ensure that the operator is trained to perform that step, and it's also going to make sure that that truly is the next step for those samples in the workflow. Now, we can get all of that information and that direct feedback from our LIMS, validating that information within seconds. So that really enables us to prevent mistakes before they even happen. So it's a really great tool and probably one of the best examples of how we implement quality by design.
Thank you for that answer. Our next question is, you mentioned that a Gemba is the best way to approach learning of a process. Don, I think this one's for you. Why do you feel it is necessary for a microbiologist to be in the production areas rather than just in the testing laboratory?
Well, I guess as I mentioned before, I believe that the best way a microbiologist can accomplish their objectives in developing robust microbiological control strategy is by being active. And an active microbiologist, to me, knows what's going on in the production area rather than just accepting samples in the microbiology lab. I'm more from the viewpoint that instead of accepting outcomes from a black box, like getting a brand new instrument in the laboratory and just expecting it to produce whatever results and then dealing with the results, my thought is that you need to go back and understand why the instrument works, what are the principles of the instrument doing what it needs to do, and what are the impacts of samples going into that instrument. And the same thing goes for a microbiologist in the production facility. If we're intending to help maintain and oversee control for microbiology in a production facility, we need to understand the processes and the people and what impacts they're going to have on that. So that's why, to me, a Gemba approach works best. And it doesn't mean that you have to specify and say, okay, I'm going to go out and do a Gemba. In a lot of cases, any laboratory that has people that sample from the production area or sample from water systems or sample anywhere in the environment of a manufacturing facility are actually performing a Gemba while they're sampling, because they can see and hear what's going on out there, and they understand what kind of impacts that can have on the samples that they're taking.
Thank you for that. Our next question. A suspected member of the B. cepacia complex is found on the selective medium BCSA. What identification method would you recommend to speciate the isolate?
So this is a really great question. This is about Burkholderia cepacia complex, which, as I mentioned during the presentation, it's increasingly of regulatory interest, which is why USP is writing a chapter. It could be USP 60. Burkholderia cepacia is responsible for a huge number of recalls, in particular for non-sterile aqueous products. And keep in mind that we call it, Burkholderia cepacia complex. That's because it's actually made of about 20 different organisms that are so closely related, it can be very difficult to speciate them. You are not going to get speciation through typical IV methods. If you truly want to know which member of the complex is being recovered in your facility, you really have to go to a more specialized service. DNA sequencing, because it uses the 16S rRNA gene, these are so closely related that that gene is not sufficient to give you the discriminatory power to speciate. You're going to have to start sequencing other ancillary genes that are housekeeping genes that have more variation in them in order to truly determine which single species is available. Now at Charles River, we do have this ability to speciate BCC. This is part of our ProSeq service. And again, what we do is we sequence a different gene housekeeping that has increased variation that will allow us to determine which exact organism you have. And this is important because there's different levels of pathogenicity between the different species that make up the Burkholderia cepacia complex. So I highly recommend sequencing, and again, it's going to need to go beyond just your typical 16S. You're going to need to do something like the ProSeq service.
Well, thank you for that, and thank you for your questions, everybody. Before I finish the webinar, I would like to ask our presenters if they have any closing remarks. Don, do you have any closing remarks?
I'd just like to say I'd like to thank everyone who's listening and participating in this webinar today and using the webinar in the future if you're able to access it, because we're all interested in continuous learning in microbiology, and you're all my peers, and you all understand how important it is to share experiences and knowledge with each other. And together we can hopefully develop and design safety into all our products.
And Jessica, do you have any closing remarks?
I want to echo what Don said and thank you all for your time and attention. I hope you guys really saw the connection between our presentations. Putting in an overall quality, environment, even within microbiology, that same quality has to extend to your microbial identification. Because again, you're making manufacturing decisions based on what you're recovering on the manufacturing floor. So thank you again for your time and attention. This is a really important thing professionally, but personally as well, as I shared that, I have such a big commitment to patient safety. So thank you for all of your steps in making sure you have quality products.
I'd like to take this opportunity to thank our presenters. So thank you once more, Don Singer and Jessica Razor, for sharing your knowledge with us. I would like to remind our audience that they can view this webinar and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send in your questions over to me at stephen.edwards@biopharma-asia.com. Thank you, everyone. Have a great day.